Meiosis, a specialized form of cell division that is essential for sexual reproduction. Meiosis reduces the chromosome number by half, creating four genetically diverse haploid cells from one diploid cell. This process is key to genetic diversity in organisms that reproduce sexually. In this video, we'll break down the stages of meiosis, providing insights into how it ensures that offspring have a unique combination of genes. Meiosis consists of two sequential divisions, meiosis 1 and meiosis 2. Each of these divisions has its own distinct stages: prophase, metaphase, anaphase, and telophase. Together, they transform a single diploid cell into four haploid cells, each with half the number of chromosomes of the original cell. Let's begin with meiosis 1, which is often referred to as the reduction division because it reduces the chromosome number by half. The first stage of meiosis 1 is prophase 1. This is a complex and crucial phase where several important events occur. Chromosomes condense and become visible. Homologous chromosomes, pairs of chromosomes that have the same genes but possibly different alleles, pair up in a process called synapsis. The paired chromosomes form structures called tetrads or bivalents. Crossing over occurs, where the sections of DNA are exchanged between homologous chromosomes. This recombination creates new combinations of alleles, contributing to genetic diversity. Additionally, the nuclear envelope breaks down and the spindle apparatus begins to form, preparing the cell for the next stage. Next, we move to metaphase 1, where the homologous chromosome pairs, or tetrads, align along the metaphase plate. Unlike mitosis, where the individual chromosomes align, here the pairs line up side by side. The spindle fibers attach to the centromeres of each homologous chromosome, ensuring that each pair is ready to be separated. During anaphase 1, the spindle fibers shorten, pulling the homologous chromosomes apart toward opposite poles of the cell. This separation reduces the chromosome number by half, with each pole receiving one chromosome from each pair. Importantly, the sister chromatids remain attached at their centromeres during this phase, setting meiosis apart from mitosis. The final stages of meiosis 1 are telophase 1 and cytokinesis. During telophase 1, the chromosomes reach the poles and the nuclear envelopes may reform around them. Cytokinesis follows, dividing the cytoplasm and forming two haploid daughter cells. These cells have half the number of chromosomes, but each chromosome still consists of two sister chromatids. With meiosis 1 complete, we now enter meiosis 2, which is similar to mitosis but without another round of DNA replication. Meiosis 2 will separate the sister chromatids, ultimately producing four haploid cells. The first stage of meiosis 2 is prophase 2. In each of the two haploid cells, the chromosomes, each still made up of two sister chromatids, condense again. The nuclear envelope, if it reformed during telophase 1, breaks down and new spindle fibers form, preparing for the next division. During metaphase 2, the chromosomes line up individually along the metaphase plate, just as they do in mitosis. Spindle fibers from opposite poles attach to the centromeres of the sister chromatids, ensuring they are aligned for separation. In anaphase 2, the centromeres finally split and the sister chromatids are pulled apart to opposite poles of the cell by the spindle fibers. Each separated chromatid is now an individual chromosome. This stage ensures that each resulting cell will have the correct number of chromosomes. The final stages of meiosis II are telophase II and cytokinesis. During telophase II, the chromosomes reach the poles and nuclear envelopes reform around each set of chromosomes. Cytokinesis then occurs, dividing the cytoplasm and creating a total of 4 haploid cells. Each of these cells has a unique set of chromosomes, thanks to the processes of crossing over and the independent assortment of chromosomes during meiosis I. Independent assortment refers to the way these homologous pairs are oriented at the metaphase plate. The orientation of each pair is independent of the others, meaning that the maternal and paternal chromosomes are randomly distributed into the daughter cells. This randomness plays a crucial role in generating genetic diversity, as the combination of chromosomes that end up in each gamete is different every time meiosis occurs. Meiosis is crucial for sexual reproduction, generating genetic diversity in populations. The haploid cells produced by meiosis are gametes, sperm and eggs in animals, or pollen and ovules in plants. When these gametes fuse during fertilization, they restore the diploid chromosome number and combine genetic material from 2 parents, contributing to the genetic variation that drives evolution.